I'm really looking forward to our conversation. Should we kick it off?
Yes, let's get it rolling. So
All
hello,
right.
everybody, and great to have you all here today. We're going to have a great chat between Doug, a managing partner at Mission Bio, and Dr. Nikole Kimes, who I will introduce briefly. I'm Reshma, visiting partner at Y Combinator, and hopefully today we plan to just have an informal chat where Nikole, Dr. Kimes, so is going to tell us about her company, and then we'll also take some questions from the audience. Um, so Nick, Dr. Nikole Kimes is founder and chief executive officer at Schulte, a San Francisco-based biotech company, co-founded with Dr. Susan Lynch, who is professor of medicine at UCSF, and Samir Kal, a founding partner at Coastal Ventures. And Dr. Kimes has spent over a decade of research experience in microbial ecology, host-microbe interactions, and previously supported by NSF and many other impressive both European and American organizations. So as the inventor of Schulte's technology, based on her research at Dr. Lynch's lab at UCSF, she's provided the foundation for the translational research program at Schulte, which she will tell us about. So we're really lucky to have her here today. And Doug and Nick, take it away.
Great.
All
So
right.
I think you're going to start, right, Nick, and tell us about Schulte.
Yeah, I believe I have a 2-minute intro.
Take your time. Take your time.
Yeah, we are a clinical stage biotech company based here in the Bay Area. We are a spin out from UCSF, as Reshma told you in the introduction, and we are really on a mission to transform the way that we approach chronic diseases. We're doing that through the incorporation of a new technology through microbial-based therapeutics, as well as integrating somewhat of an innovative approach to chronic disease through early intervention. So what we're bringing to the table is really a deep knowledge around the developing gut microbiome in the early years of life, and combining our knowledge of microbiology, immunology, and bioinformatics to develop targeted microbiome-based therapeutics and accompanying diagnostics to really go after targeted treatments of chronic disease. We're starting in the realm of the atopic march, and we really see a future where we can alleviate this progression of allergic diseases. It starts with atopic dermatitis, and then you see development of food allergy, and eventually it moves through to allergic asthma as well. And so our first indication is all of those allergic diseases wrapped up as one, and we're very interested not only in treatment, but in the actual prevention of this march through early intervention. That's the big nutshell version.
Okay, but I wonder if you could go one step further, because this is such a wacky idea that gut microbiome determined asthma years later. Tell us a bit more about that and how you got to that idea. Yeah.
Well, we know that it started with an association and trying to figure out what were the risk factors for allergic diseases. And the things that were well established were things that impact early life. So the mode of birth, whether you're feeding breast milk or formula milk, introduction of antibiotics to a pregnant mom or to the infant early in life. Another drug that's actually associated with high risk is also the anti-reflux drugs. And all of these risk factors are known to also impact the gut microbiome. And so this is where Sue started over a decade ago of trying to figure out what was it about the microbiome. And we know that the first year of life, the microbiome is absolutely essential in training the immune system and developing immune tolerance. And that's where the body gets used to the commensal organisms that are such an integral part of our human ecosystem. And these risk factors are known to really cause disturbances to what we think of as the typical progression of microbial development over the first year of life. And so trying to figure out what was happening once the system was disturbed. And so for us, once we knew that the microbiome could predict the kids that developed disease, the question was, could we prevent it using those same microbes? So we spent a lot of our early years, and certainly in Sue's career, doing comparisons between, in infant cohorts and figuring out not only what microbes are different, but functionally, what does that mean? How does that impact the functional capacity of the human ecosystem? And that was where we really started to be able to build on that to design microbial consortia that can replenish the functionality that's missing in those high-risk kids that go on to develop disease.
So I can totally get how you could be training the immune system in a way that would be both helpful and unhelpful to a child and adult. But you have also found that introducing appropriate microbiomes later can have salutary effects, right? So that's not about
Absolutely.
training. Yeah, so there's got to be some ongoing communication that's occurring there.
So training is the way we think of it if we really want to prevent disease from happening, right?
Yeah,
Because
yeah.
that's getting in early and making sure that those shifts don't happen. Once those shifts have happened, the microbiome is still an essential part of immune functionality as well as metabolism. We know one of the really exciting things to me about live biotherapeutic products, which is what a microbiome-based drug is classified as, is that they are these really complex, dynamic, and integrated medicines. So they have, rather than thinking about a single pathway with a single mechanism of action, we're really looking for these complex multiple mechanisms of action. So, we know that microbes have cell-to-cell interaction with certain immune cells. That's one way they can impact immune function. We also know that they metabolize complex structures into to metabolites that then circulate throughout the system. And this is another mechanism of action. We also know that the microbes impact epigenetic responses in the human body and human cells as well. So there's an epigenetic component. There's also IgA antibodies that are stimulated by the microbes. So there's all of these different pathways that can be utilized by these organisms and through these drugs that allow us to start thinking about really having a suitable answer to complex, intricate, chronic diseases where you have redundant pathways.
So I should also confess we have three purposes today. One is we'd love to learn about Shilta, which we're doing right now, and we're going to do more of that. A second is, and actually I'll also say two purposes. So that's one. The second is that we'd love to help inspire entrepreneurial scientists who are contemplating creating life science companies like Shilta to take the dive, to go ahead and do it. So we're here to inspire them to leap. And part of that, and this is sort of the third part, is you're such a beautiful example of that path from basic science through to successful biotech leadership, that your story, you as an individual, will be the inspiration for those that follow. So I wonder if we could turn a little bit from Shilta's science, which we'll get back to. I'd love to learn more about that. And just ask you a bit about yourself and how, and the arc of your life that brought you to science and then to Shilta. I wonder if you'd riffed on that.
Sure. Reshma gave me some questions earlier, and one was, tell us about your childhood, which I have never really thought about how to place my childhood in the streamline of what has happened since then. But it was really interesting to think about. My mother's a nurse, and my dad was he went between finances and business, and at some point they actually worked together and ran an emergency healthcare center.
Uh-huh.
And I never thought about it, but that was the first business I was exposed to. What I remember is the science part. I loved going, and they would let me spin down the blood samples or help take x-rays, or I was 9 years old in a doctor's office running different machines, and I loved it. So I always had a tendency towards science. I think I started with like many people do. I wanted to be an astronaut and a pilot, and so more engineering, hard sciences. And then as I got into college, really moved more towards the biology, and then eventually towards complex living systems and environmental science. And I've always had this desire to bring environmental science and health with human health. And that's really what most of my trajectory has been, is sort of bouncing back and forth between human health and environmental health. And I have finally gotten to a place where I believe we're finally at the point of acknowledging the intersect between those two, and how the microbiome is the key modulator between environment and human physiology. So it's been a great meandering path to get here, but I have found my happy spot.
And where'd you do your undergraduate and graduate work?
I did my undergraduate at the USC, the original one, University of South Carolina. And then actually came out here to California and did a master's in holistic health education, which is where I really cemented my love of the environment and sort of systems biology, living systems. And then I ended up doing my PhD at the Medical University of South Carolina. And again, that was me. It was an environmental science program that was at a medical school. So by definition, we had to find a way to tie it all back into human health, which is what I was looking for specifically.
And then you did a postdoc with Susie, I take it.
I did. So I actually, after finishing my PhD, a bit of an adventurer and needed to do something outside of the US. That was, my husband and I had agreed on that. So I went to Spain and did a postdoc, which was phenomenal. And it was in metagenomics, really a lot of the bioinformatics side. And then from there came back to the Bay Area and specifically sought out Sue, because she was one of the only people in the field of the human microbiome that was very obviously and vociferously taking an ecological perspective. So I actually knocked on her door and told her she had to hire me, and managed to convince her to do so. And it was a great.
That's so cool. And it's wonderful to have an opportunity to go live and work overseas as part of your training. That is, and not every profession gives you that opportunity.
No, it doesn't. And it was a great experience.
And so when did you work on asthma as part of your postdoctoral work?
I did not. So I was still in the environmental side of things. It was similar. We looked at ecosystems where you have an environmental change that impacts the microbiome, and then how that affects the health of the community. But the key piece for me there in that postdoc was the bioinformatics, because all of the statistics and ecological perspectives and the multi-omics integration, that's all been being done in environmental sciences for decades. We're just starting to do it with human health. But all of the tools were actually developed by ecologists for the most part. Even sequencing, 16S sequencing was to look at soil microbes. That's, the reason why we started going down that road. So it was a great training ground for what we're doing now.
So then I wonder if you could unpack, which is where we want to help people make the leap, unpack the moments where you and Susan first started to have tea and chat about a company, or where did that come from? How did that get born?
This makes me laugh every time. I would love to say that I knew I always wanted to start a company and this was an ongoing conversation, which is the farthest thing from the truth. I had no idea. I never even considered starting a company or really even moving into industry, to be honest, until one day. It was a Friday afternoon, and I remember it very distinctly. Sue was walking out of her office, walked through the lab and said, "Hey, Nick, on Monday, I want to rethink your career trajectory. We'll talk about it next week." And she walked out the door. And I looked to my good friend Ricardo and I was like, what was that all about? And I had to sit with it for the weekend. And over the weekend, I thought, what could she be talking about? What are all the different, and it was the first time I ever thought, what are all the different possibilities outside of academia? And then we circled back around the next week, and it was really Sue who had the ideas and had always thought, we need to take this a step further and translate what we're seeing into something that's going to help patients. She always had that idea and was waiting for the right time.
Mm-hmm.
And it was a confluence of people that she had interactions with. She has worked with Mark Benioff. He introduced her to Samir at Coles Ventures. He was interested in the science. And when she was presented with the question of, well, who's going to turn this into a drug? She was like, well, surely somebody's out there doing it. And Samir came back and said, no, I've looked. Like, we need you guys to do this. Sue has made it very clear that she has no impetus to leave academia at all. So somebody had to be willing to actually go out and start the company. And that's where my adventurous spirit came in. A challenge. Yeah, let's do it. So it was really
Monday morning you said yes.
Confluence.
Yeah.
Yeah, I definitely, I never, once presented with the opportunity, I never thought twice about it.
That's so interesting. And can I presume that prior to that you had been thinking about an academic career?
Correct.
Wow.
Yep.
And so then unpack the balance. What happened over the next month or 6 months, or tell us about that process.
Yeah, it was about 6 months from the introduction of the idea where we started to think about what a business plan would look like, what money would be required, all of these things that I now take for granted that you're always thinking about, but they were so new to me. And I thought, how in the world do I put a timeline together for drug development? I don't know drug development. Like, how do I do this? And just starting to reach out to people and do research and try and figure that out. And so we put together a deck for the licensing. We started thinking about really what milestones would convince us that we had something that we should move forward. And just starting to put those little pieces together. And that happened over the next 6 months. And then I actually left UCSF and started the company in December of 2016. So I would say it was about a 5-6 month process in between there, just figuring out if we could license the technology from UCSF and how much money we would need to pull in and how we wanted to structure the company and the ownership and all of that stuff.
Is that when you moved into NBC BioLabs, or did that happen still later?
Nope, that was exactly when I moved into December 1st, 2016.
And in those first days, it was just you and Ricardo in the first instance. Is that right?
So I started it on my own.
On your
And
own,
I
okay.
was able, so yeah, the person I turned to when Sue said, let's rethink your career trajectory, was Ricardo, but it was also his last week in the lab, and he was leaving academia and going into industry. So he moved back to Georgia. So I did this early work. We started the company, licensed it, raised the money. I think we actually signed our first Series A in February. And then I was able to recruit Ricardo by March or April. So the first couple months I was on my own. I don't suggest that. You can certainly start on your own, but it really helps to have
Yeah.
a trusted co-conspirator, if you will,
Yeah. No,
in those early days.
it's a tough path. You want a team and not,
It is.
yeah.
It is
So,
indeed.
so I'm just about, I'm going to turn it over to Reshma, but there was one conversation you and I had, which I'd like to, if you don't mind, I'd like to unpack for you. I remember you came in during those early days and said, well, we're running a biotech company. We have capital. We have terrific, ambitious plans, but very daunting plans. I've been thinking about going out to get a CEO. And do you remember this conversation we had?
Oh yeah.
I said, Nick, why? You're very capable. Why don't you go until you fail, feel like you failed, and then do that? Why anticipate that you need something you don't need? And here we are some years later, and you're doing a great job. Could you tell us a little bit about that personal decision-making process and how the board felt about
Yeah.
that and all that?
That was a huge mental exercise for me. Having never considered starting a business, I just thought you had to have an MBA CEO to run a company. That was my own perception going into it. And to be honest, I would say in the people that I talked to, 50% of the people also told me that.
Mm-hmm.
But then there were the other 50%, you included, which I'm very thankful for to this day, who presented the other side, which is in startups and biotech, you have to know the science. I mean, you have to know the underlying foundation of what you're doing to really build upon that. And who better than the scientists who truly understand it and know how to build upon it. It's been an interesting evolution to me. So we're 4.5 years into this. I was not officially named CEO until we were raising our Series B. So I think I started the company as vice president or something.
Mm-hmm.
We just didn't have a CEO. And then they very slowly moved my title up. I went through CSO, and then finally moved to CEO. So it took me a long time to actually claim the depth of what I was doing. And I don't necessarily think that was a bad thing, because it also let me get comfortable with the title and what I was doing throughout that process. Now, to be very clear, I was the CEO the entire time. There was nobody else to do those things.
Right.
But it worked for me from a mental standpoint to sort of build up to that. And I'm thankful that I had the support behind me. Sue always knew that I should be leading the company. And even Samir, he felt it on your side of the equation of like, Nick, I'm going to keep supporting you as long as you can keep going. You know, once we see a problem, then let's start to fill in the gaps once we see them. But to date, we're not there.
Well, we're so proud of you, and I'm so delighted that you made that decision. You're a great role model for lots of others to follow. So thank you so much for doing that.
I appreciate those words, and it's been a phenomenal adventure, and I highly recommend it. to most people. It needs to be your thing.
But it's the kind of it's not as if you knew where it unfolded for you, right? So it's not as
No,
if we always say, if that's your thing, like you'll know in advance. Oh yeah, I'm planning on this, but you got to dive in and just do it at a certain level,
So
don't you?
let me be clear when I say if it's your thing. To me, in my mind, it is an ability to embrace uncertainty
Yeah.
and to be able to continue to function when you are uncomfortable.
Yeah.
If you have, those are qualities that I do think are necessary. You don't have to know where it's going. You don't have to know that it's for you, and know that you can always change your mind, right? That's the
Mm-hmm.
beauty of life. We can always change our path at any point in time. And just being able to embrace those elements is key.
Well said. Well said. Well, as I say, we're super proud of you. So, Reshma, I'll turn the floor over to you, and
Yeah.
we can engage the audience as well.
Thanks, Doug. And Nick, I learned a lot of interesting things in the past few minutes. Here's some of my takeaways, and maybe we can discuss them a little further. So I noticed that you've referred to a few times your interdisciplinary nature, like you're looking at environmental things in the medical school and kind of across, and this seems to be a trend in entrepreneurs. Like a lot of companies are started at the fringe. And do you see that as a theme? Is that in your experience, how has the interdisciplinary nature affected your choices in career?
I don't know if it's necessary in all businesses. I think certainly in our field, in the microbiome field, it's absolutely essential. I think one thing I do see across CEOs in seeing those that do it well and those that don't, one of the things I'm going to call it zooming in and out, this ability, like I always thought it was an affliction of mine, in that there were days where I really wanted to focus in on the details, and that was very grounding to me and made me feel stable. But there were other days where that was stifling. And what I really wanted to think about was the big picture and how this all was going to impact the world and have a benefit in this larger piece of it. And I think as a CEO, one of the things we were called upon to do is to vacillate between those things very quickly and easily, and being able to go from the detailed piece of what you're doing to the larger, broader implications, and being comfortable in navigating those. So for me, it's actually been a relief to find a role in which I get to utilize those different components. And I think of the interdisciplinary integration as a piece of that broadening and bigger picture, being able to bring that in. I think it's one of my greatest appreciations for what we're doing and what I get to do on a daily basis.
I mean, you described it as something that you learned, but I see like entrepreneurial hints in the description that you've given throughout your life, like a 9-year-old with the confidence to take x-rays and do spin down blood. I certainly would not have done that. Or having the confidence to ask Professor Lynch for a job and make her hire you. I mean, that's like a entrepreneurial.
Interesting to hear it put that way, because I never thought of myself as an extremely confident person. And certainly internally, I feel a lack of confidence often, but it's never stopped me from trying something.
Yeah.
So whatever that feeling is, I'm willing to push past it.
Yeah, maybe that's a note for all those in the audience. Like, even if you feel some doubt, like everybody does, and maybe you can consider taking the plunge. I mean, it sounds like Nick got a call on a Friday and was like by Monday interested to start a company, and her opportunities were open. But I'd like to drill in a little bit to your discussion on milestones. You mentioned like you guys started the company in maybe end of year, December 2016, licensing, discussion, etc. Series A in February 2017. Can you talk about like in a little bit high res, like what were you licensing and kind of what was the contracting process? And then what evidence did you have to like raise your Series A?
Yeah, so I know so much more now looking back on it in the way I think of it. Starting, I was like, we're just an academic lab. We just have preclinical data.
Mm-hmm.
Preclinical data is really good data to have.
Well,
and I think we started with a lot. Yeah. What was that?
What was the nature of the data? Was it like, you know, infant samples from 50 patients, or what was it?
No, so we had, you know, Sue's previous work had used clinical data from infant cohorts to differentiate between high risk and low risk infants. So the preclinical data that I worked on was actually designing a microbial consortia based on those differentiators, and then going into animal models and seeing if we could actually alleviate airway inflammation through oral supplementation with the microorganisms. So we had some preclinical animal data.
Pause for a second. So I'm like always the least educated person in these conversations. Okay, so by the consortia, it's like you have like a census of all the different organisms, and then your data was if you.
Yeah, a consortia for us is just a group of organisms. So yes, we had, you know, it started with a whole list of what's different between the two. And then you narrow it down to functionally which ones are playing the biggest part. And then you can narrow it down even more based on starting to understand the individual characteristics of different organisms. And then, you know, that gave us a group of organisms that we could manage in a lab at the time, right?
So you have preclinical data on different consortia and what effects they had on mice. And that was the core IP that you licensed? And are these like naturally occurring consortia, or like are engineered in any way, or?
They are not engineered. Naturally occurring organisms found in healthy humans.
Awesome. Okay, so for the audience, this was kind of the nature of the data that needed to be licensed from. I mean, can you even, what were you licensing? Can you even license naturally occurring organisms?
So you can license anything from a university. You're asking whether you can patent those organisms. So
I'm wondering why you needed like a licensing deal from UCSF if like those are naturally occurring organisms. I'm truly uneducated, so I just don't know.
So this is a whole IP conversation that I could spend hours on now, which another thing I never knew I'd ever do. But it's the patent side of things. Most people think you cannot patent naturally occurring organisms. You absolutely can, and the oil industry has been doing it for decades. That being said, it's harder now. You can still patent organisms through different measures if they have different functionalities, if they're being combined in an unnatural way. Also, if you can show evidence, which in our case, what we focused on is that they're synergistic. So if you look at the functionality of each organism separately, it's not the same as when you put them together. When you put them together, they have a synergistic impact. And so that is a patentable approach. And then you can get into whether you're you're getting patents for the composition of the consortia, or is it for the method of use that you're actually using them to prevent disease, or you can go about it in many different ways. But the licensing, which is different because that's coming from the university, anything that was developed at the university they have ownership to. And if you want to use it in any meaningful way, getting that license so that you can then control the patents and control who else has access to it, then you need to license that from the university.
So you were licensing like the specific mix or the process to make the mix from the university?
It was a bunch of those things.
A bunch of those.
Mix itself, the methods to design the mix, and then as well, we're also interested in diagnostics and the methods for using this type of data to develop diagnostics as well.
Got it. And did they, like, you mentioned this was before you'd raised your Series A, so did you have to pay them to license, or do they take like some shares in the company? How does that work?
You can set it up in many different ways.
Mm-hmm.
Typically there is a small upfront fee and a yearly licensing fee associated with it. But the idea is to design the licensing through milestones where they'll get paid downstream as you're successful in development. You don't want to make it upfront heavy, but typically there's some fees associated with that. And it was one of those where we had people interested in financing us, but they weren't going to finance us unless we licensed the technology. And then to license us the technology, they want to know that we're going to have the financing to develop the license. So it was really trying to coordinate those two things in the beginning.
Yeah, how did you break free of that bind? Because that actually seems like a little bit of a nut.
Communication and bringing everybody to the same table.
Mm-hmm.
Like, here's what we want to license. Here's people who are interested in helping us develop this. Can we all agree that we can make this work?
Yeah.
So we actually brought our investors into the licensing conversation.
Oh, wow. Okay. They're kind of like, you got some interest from some investors and then you took them on the journey with you as you developed the licensing plan.
We did. Yeah. Which is a pretty unique way to start a company. I guess I don't know across the board, but it's not the same as somebody who starts their company, license their IP, and then goes out and looks for money. That would look slightly different.
Yeah.
Same process, just not integrated up front.
Totally. I need to dig up the post, but I'm working at YC now, and we've put online some templates for licensing agreements with the university to just get the conversation started if you have a patent office at your university. But I think Nick was on the frontier, really figuring this out ahead of time before it was a very well trodden process. So pretty interesting. All right. So and once that was pretty much all sorted out, you were able to raise your Series A, get started on building technology, and you the IP that you've developed between the Series A and the Series B. Can you kind of give us a picture, if there was a pie chart, what slice is the original and what slice is kind of new from your development process that you've been doing in the interim?
Yeah, that's a hard pie chart. The technology that we took into it is really foundational to everything that we've done. So I have a harder time putting that in a piece of the pie.
Yeah.
But it was really the concepts, the licensing, and the early preclinical work was, that initial what was coming to the table. Between Series A and Series B, it was putting together a team, having a lab, and just showing operationally that we could function. Preclinical validation. So we, had to rerun everything that you're doing in academia. Not exactly, but just to show that in different hands that you're going to get similar results, and that it's not something that was unique to that lab space. And then for us, because we were in the microbiome space, which is new, really figuring out our trajectory between, are we a food, are we a medical food, are we a drug, and figuring out our regulatory route was a huge piece. We chose to go the drug route very specifically. And because of that, the next milestones were really around regulatory, getting an investigational new drug application accepted by the FDA. And then manufacturing is another piece for us. Again, sort of unique to the microbiome space, that there's not manufacturers out there that you can just go to and say, please make me these really strict anaerobe organisms that you've never grown before. So we developed manufacturing in-house. And we had to develop it at a GMP level so that it could be used in FDA regulated clinical trials as a drug.
Can you educate us a little bit? Like, is this a biologic or is it like a small molecule? Like, as I'm trying to think about the categories.
Yeah, I definitely think of it as a biologic. So it first and foremost is a drug. Then it's a biologic. So we actually are regulated through CBER, which is the Center for Biologics, not CDER, which is for small molecule drugs. And then within CBER, we very specifically are under, it's the Office of Vaccines, which we are not a vaccine. But we were told by the FDA in one of our conversations, which I thought was really progressive of them to think of, but we were told that it was in the Office of Vaccines because that is their most sort of out of the box thinking program. And they knew the microbiome space was unique and was going to need a unique focus from the FDA's perspective. So they specifically put it in biologics under vaccines so that they could have more flexibility in how they manage program to program.
Whoa, crazy. Okay, that's really interesting. Okay, I'm going to ask one more question. I'm also going to remind the audience that we'll be taking questions, and I'm going to paste in a few blog posts. We have some materials on this topic for those who are interested while you type in your questions. But final question from me before we open up to the audience. When your Series B was, when you were raising it, like what kind of 4 points did you make as part of your pitch that you should get Series B funding? Like what milestones scientifically had you met, and then also with your team and the business?
Yeah, you know, it's the really traditional venture capital bullet points. We had a extremely knowledgeable and productive team. We had set and hit all of the milestones put before us over the last 2 years. We had de-risked the technology in that we were able to do the manufacturing and have product that we could move into clinical trials, particularly into phase 2 clinical trials. And then really the regulatory path forward. It was really important for us specifically because we're interested in early intervention. So moving into infants and children and showing that that's possible, because typically you develop a drug for an adult and then move into children. We're doing it the inverse, where we're starting with the children and then moving into the adults. And interestingly enough, the FDA is helping us on that end, in that they're really starting to recognize that drugs for children are different than drugs for adults, and that it shouldn't just be a sliding scale down, that infants are unique. So they're actually embracing that process a little bit more. But I would say those are the 4: the manufacturing, the regulatory, the team, and then just operational efficiency, that we can do what we say we're going to.
So cool. I want to just comment a little editorial here. Like the FDA hasn't approved a new manufacturer for infant formula for like 100 years. So some really, really long time. So this is quite a feat, and I could see how
Keep
it
your
could.
eyes open. I think that might change very soon.
Oh, all right. I would. Well, maybe that's an after discussion for us. But
Yeah.
cool. Let's see. So we have a few from Carissa. How did you navigate through the business side of starting a company, especially at the very beginning? Did you work with a team of experts, or did the majority of the effort fall on your shoulders?
Most of it fell on my shoulders, and I'm going to give a huge shout out to NBC BioLabs here. And this is more just from a logistical standpoint of like, what licenses do you need? What state authorities do you talk to? What insurance do you need? I knew nothing about any of that, right? And I walked into NBC BioLabs and Doug gave me a checklist. Well, actually, Linda gave me the checklist.
Yeah, Linda would. Yeah.
And I really, this is something I would say to all entrepreneurs, and I don't mean to be a commercial here, but it's really overwhelming and isolating in the beginning, and having a community like Y Combinator or NBC BioLabs, just where there's other people navigating these processes, was invaluable. Very much so.
I hear that. And I mean, Doug and I are putting these events together actually to kind of let people know that there's a community there who is like supportive on that front. And we're really like, if you have the scientific firepower to skills to start the company, that there's support on this side, if that's at all interesting. But, cool.
Yeah.
Question 2, once funded, do you try and create IP outside the university so you don't have to license through them?
You are trying to create IP as much and as often as possible. Absolutely. Now, sometimes you're not going to get around your founding IP, and that's always going to be part of what you do, or at least for a good portion of time. But yes, you definitely want to be trying to create additional IP that is independent of the universities.
Yeah. And do you have like a IP, you're continually filing like lots of patents and provisionals and stuff? Is that kind of your strategy?
So this is an interesting piece, and part of this was discussions with Doug early on. Part of this was discussions with some other advisors as well. I was told early on that if I was going to spend money, IP was one of the areas to spend money, it specifically around drug development, that it was going to be very important moving forward. So that is where I've probably had the most input and spent a lot of capital working with a firm that drives our IP.
Totally makes sense.
In a very, we pay a good price to have a well-recognized representation in that area.
Not cheap.
And it has paid for itself for sure. It's an important piece. So I would continue to reiterate that is one area where you do want to invest some money, because if you don't do it right, it can really hurt your chances of moving forward.
Yeah.
So what's the next big thing in the microbiome outside of consortia? Some examples: bacteriophages, CRISPR, bacterial editing, or evolution. Not sure what that means, but maybe you do.
I think there is no end of answers here. I think we are just learning and becoming knowledgeable and tapping into the microbiome. We've really spent the last decade just getting people to know that it's a thing. But with this recognition, now you have lots of different fields that are focusing on it, whether it be your phage or your AI or your health tech. There's so many different areas now that are starting to incorporate the microbiome. So I think there is no end of opportunity here. There's so many different places. I do think that the engineering side of microbes is really interesting to me. I know people were afraid of it initially and thought the FDA would never support engineered microbes. The FDA loves it. If you can engineer a microbe, you can also engineer it to kill itself or turn itself off. So they actually like it and think it can be easier to control than something that's naturally occurring that belongs there. So there's tons of opportunity in this space. And
this is the beginning of every sci-fi movie anyway.
I'm thrilled to get to watch it happen.
Let's see. And two more quick ones. Maybe we can do them lightning. Does your company have a continuing relationship with academic lab of origin?
We do. So Sue is on our board. She is also an advisor. This is important for us in the initial focus that we've chosen for our indication. She's, a leading KOL in this space, and to have her voice at the table is important. It does complicate things. We have to be very careful about conflict of interest and what she's doing in her lab versus what the company does, and it can cause some hurdles that we've always figured out how to manage them and keep everybody comfortable in the way that we do it. And it's been quite beneficial for us, just something that you have to really pay attention to, how you set that up.
Yeah, makes sense. And then final one, how much IP do you need to start a company, especially in the context of talking to students? Maybe a big question, but from your perspective.
Yeah, I would definitely defer to the VCs on that side of things. I mean, for us, it was just having a license to the foundational technology. That's what we had to have to get funding. Without that licensing, we would have never gotten funding. So we did have to start there.
I mean, I would encourage people also, I'm just Reshma at Y Combinator. We do if people have questions about whether they're nugget of stuff they've been working on, their thesis is licensable or the whole dynamics of that, we do some discussion on it if you're interested to have a discussion. It is, I think, like to just see what I compare Nick's case where she went from university direct to Series A. Now we're seeing a little bit more of a progression, people leave,
right.
They raise a small round, develop some small prototype, and then move on to A. So there's a, more smaller steps, which is a additional option.
I would say in trying to figure that out, don't be afraid of the investors. Everybody's afraid of investors. You can use them just as much as they're going to use you. They are resources who will tell you how much IP they want to invest in you. So go ask them. It doesn't hurt to ask.
Yeah, and just to add a story to it, so one of the companies in our community started, and they came to us for funding, and we said, "Oh, what's your IP?" And they said, "Oh, we don't have any IP." And we said, "Oh, what's your product?" "Oh, we don't have a product." And we said, "Great, where do we send the check?" And that company now has 800 employees, and it's raised $800 million. That's Zymergen. And so, yeah, don't be afraid.
Totally. There's almost no penalty for trying many times, very low penalty. Applications are open for YC, and please feel free to reach out if you even have a thought that it could be a business. I completely agree with Doug. It's it's not like there's some rubric that is gonna just make things happen. Every case is unique. So we are at time. But thank you, everybody, for joining today, and especially Nick for sharing your story and dealing with our questions. And hopefully, if there's folks in the audience who are interested in starting a company, or you have colleagues or friends you've worked with in the past who have interest, we'd love to connect with them and make sure that they have access to the resources that would help them. So thank you, everyone.
Nick, thank you so much. You were great.
Thanks, Doug. Thanks, Reshma. I appreciate the opportunity.
You have a wonderful day. Bye-bye now, everyone.
Bye, everybody. Good luck.